Okay, hey guys, I'm Judy, and I'll be taking you through the main mechanisms of antibiotic resistance and briefly go through how this can spread from one bacterium to another. So, of okay. Starting off with the modification of the antibiotic target, there should be a diagram that comes up. Okay, so this diagram shows us how bacteria become resistant when a mutation changes the antibiotic's target site. So I'll give you a named example. I'm sure you've heard of MRSA, which is a resistant form of the bacterium Staphylococcus aureus, and this can resist the antibiotic penicillin. So I'll just give you a bit of background on penicillin. It's an antibiotic that targets the bacterial cell wall, which is made up of a polymer called peptidoglycan. So I want you to remember me on that one. So when penicillin is used against a normal antibiotic-sensitive bacterium, when penicillin is used against a normal antibiotic-sensitive bacterium, it attaches to a penicillin binding protein, and this is an enzyme that helps build the cell wall. This will then block the usual peptidoglycan molecules from attaching, so the cell wall can't form properly since it's made up of a monomer, or of a polymer, which is like many monomers joined up together, so then the bacterium dies. However, if you see the diagram, it shows that after a mutation, the shape of the binding site will change, and this means the antibiotic can no longer bind, but the peptidoglycan substrate can still attach. So this means the cell wall forms normally and the bacterium survives. And this also shows that it's resistant. Moving on to the other mechanism. Okay, so we have preventing the antibiotic from reaching its target. So instead of changing the target itself, some bacteria stop the antibiotic from even getting to where it needs to work. So the picture should come up. Okay, so if you tap one more time, it's going to show where. Okay, so the first one, there are some bacteria that produce enzymes that break down antibiotics before they reach their target. Okay, so I'll go more into this later because it comes under destroying the antibiotic and modifying the antibiotic. Tap one more time. Okay, so there are some bacteria that use efflux pumps. So these are just proteins in the cell membrane that actively pump antibiotics back out of the cell. So this happens before they can even reach their target. And then finally, we have decreasing the uptake. So bacteria can reduce how much antibiotic enters the cell. And this, again, is called decreased uptake. So, for example, there's gram-negative bacteria like E. coli that can change the proteins in the outer membrane, and this makes it harder for antibiotics to enter. So after that, I'll just go more into destroying the antibiotic. So it's going to show another diagram now. This is just a general structure for antibiotics that contain a beta-lactam ring. This is shown in pink. So there are some antibiotics that synthesize enzymes called beta-lactamases, and these hydrolyze the beta-lactam ring. And this means that the antibiotic will change shape. So if it changes shape, it means that it will no longer bind to the specific target that it has on the bacteria. So it doesn't kill the bacterium, and then it just shows again that the bacterium is resistant. Then we have modifying the antibiotic. So if you looked closely on the diagram before, there was an antibiotic with an attached molecule on it. So if you click one more time, it's going to show streptomycin. This is an antibiotic with the hydroxyl group at the bottom highlighted. This is because the hydroxyl groups on this antibiotic are very susceptible to change. So we could have enzymes attaching phosphate groups, acetyl groups, and again, it follows the same mechanism. So the shape of the antibiotic changes, so it no longer binds to the target on the bacterium. And again, that's another mechanism of antibiotic resistance. Okay, and all of these occur because of mutations. So mutations are changes in the base sequence of a gene, and these genes code for proteins. Next. How antibiotic resistance spreads. So we said that these mutations cause the resistance. There's the vertical gene transfer where the parent. Wait, okay. This just shows binary fission. So binary fission is the method by which a bacteria divide. So from the parent cell, the mutated gene spreads, and then we have the other offspring bacteria with the same gene. Next, we have, I'll just briefly go through the horizontal gene transfers. So this is a plasmid with the resistant gene on the side. Then we have conjugation of the bacteria. So if you see on the diagram, there's a pilus, and this is just the bridge between the two bacteria. And then the plasmid is copied and shared between the two. And this is transformation. So DNA fragments in the environment can be taken up by bacteria, and then this fragment is then incorporated into the bacterium's genetic information. So then it produces different proteins that can be used by the bacteria in resistance. Next. And then this is transduction, which is used by viruses.
Right, so I'll be talking through causes of antibiotic resistance. Antibiotic resistance develops when bacteria gains genetic changes that allows them to survive drugs designed to kill them. This can occur through genetic mutations, which are random DNA changes that may alter antibiotic target sites, such as ribosomes or cell wall enzymes, so the drug no longer binds effectively, or through horizontal gene transfer. The bacteria share resistance genes via conjugation. This is when direct plasmid exchange occurs between cells. Transformation, absorbing free DNA from the environment, or transduction, which is gene transfer by bacteriophages. These are viruses that infect bacteria. Misuse or overuse of antibiotics creates selective pressure, meaning susceptible bacteria die while resistant ones survive and multiply. Resistance mechanisms include enzymatic drug inactivation. For example, beta-lactamases break down penicillin-type antibiotics. Efflux pumps that actively expel antibiotics. Reduce membrane permeability, preventing drug entry, and metabolic bypasses that allow bacteria to continue essential processes despite antibiotic action. Poor infection control, agriculture antibiotic uses, and global travel further accelerate the spread of resistant strains.
Hi guys, my name is Anish. So I'm going to talk about the global impact of antibiotic resistance. It's recognized as one of the top 10 public health threats by WHO, and it's listed as a chronic risk by the UK. In 2019, infections resistant to antibiotics claimed at least 1.27 million lives, and it was also associated with nearly 5 million deaths. It's also estimated to currently claim at least 50,000 lives each year just across Europe and the US. There's also economic repercussions too for healthcare systems like the NHS, for example, and also economies. For example, it creates need for more expensive and intensive care and affects the productivity of patients or their caregivers because of prolonged hospital stays. It's definitely a problem for all countries at all income levels, but those living in low resource countries are impacted the most. The spread of antibiotic resistant infections obviously doesn't recognize things like country borders. So in these low resource countries, there are contributing factors such as the lack of access to clean water, or poor infection control, or poor access to vaccines and medicines and things like that. So that was the current impact, but we also have projections for the future on how antibiotic resistance can affect us. So by 2050, antibiotic resistance could be attributed to 10 million deaths every year, according to a review of antimicrobial resistance. From the graph you can see on the right, for context, currently 8.2 million people die each year to cancer and 1.2 million to road accidents. As for the economic projections, the World Bank estimates that antibiotic resistance could result in 1 trillion in additional healthcare costs by 2050. And the review estimates that it could cost the global economy up to 100 trillion if left unchecked. So despite the size of all those figures, they don't really capture the full picture. One of the greatest worries about antibiotic resistance is that modern health systems that rely heavily on antibiotics could be severely undermined. For example, patients are given antibiotics before most surgeries to reduce the risk of bacterial infections. So in a world where antibiotics don't work, surgery becomes far more dangerous. Chemotherapy, for example, becomes a lot riskier as it suppresses the immune system, and antibiotics are relied upon to protect them from infections that their immune system can't fight off alone. So these things are why medical professionals consider these secondary effects the greatest risk posed by antibiotic resistance. Potentially, these medical procedures that have saved millions of lives could be undermined, which is why the world has to do something about antibiotic resistance. And yeah, that's all I have to say. Thanks.
Hi, I'm Aditi, and I'll be talking to you about the Antimicrobial Stewardship Program. So, antimicrobial stewardship is defined as an organization or healthcare system-wide approach to promoting and monitoring judicious use of antimicrobials to preserve their future effectiveness. So this is really useful because, as we've seen, there's many negative impacts that antibiotic resistance can cause. And a way to combat this is by making sure that drugs are used in the right way and not used when they're unnecessary. So the main areas that the program targets are called the 5Ds, and they help make sure that antimicrobials prescribed are suitable for use. This is in response to research that showed that roughly 20% of prescriptions in antibiotics in primary care in the UK were incorrectly prescribed in 2018. So yeah, there's the 5Ds of antibiotic stewardship are diagnosis, drug, dose, duration, and de-escalation. So when you're diagnosing illnesses that require antibiotics, you should make sure that you do make the right diagnosis based on actual information, such as like sending cultures to the labs before you prescribe someone the antibiotics, and making sure that the drugs used are correct, the dose is correct, and duration as well, because a lot of people don't end up taking the full course of their antibiotics, which can also lead to further problems. And de-escalation, making sure that you review when antibiotics are given out every sort of 2 days, so that you don't use antibiotics when they're not necessary. Next slide, please. So in healthcare systems, antimicrobial stewardship is promoted mainly by people who are higher up in the sort of the NHS and big healthcare systems. And this can happen through education, through things like virtual courses. So increasing the education across healthcare professions makes sure that everyone knows the impact of antimicrobial resistance, just as you guys do today. so that they can think up new policies and stuff that could help in the future. Increasing education amongst the general population also helps to understand why their doctors may not be prescribing them antibiotics and the alternative treatments available. Prescribing antimicrobials for suspected infection only when biological samples from the patient have been sent for further investigation. So if they've been cultured in a lab and it comes back that they do have bacteria and it's not self-limiting, antibiotics should be prescribed. Recording data from instances where antibiotics are being prescribed to allow for tracking of local patterns of resistance. Re-evaluating whether there is a need for an antibiotic prescription by addressing the needs of patients via consultations. Following medical guidelines, such as the NICE antimicrobial prescribing guidance for determining the duration and the dose of antimicrobials. Updating policies according to new guidance to help manage antimicrobial resistance and implementing the strategies in the healthcare system. And better hygiene and sanitation in facilities to prevent the spread of infection. So yeah, some infographics to summarize that. And the bottom picture is the NICE prescribing guidelines. So the benefits of the antibiotic stewardship program are that over time, antimicrobial resistant infections should decrease if these things are implemented. And there may be a significant decrease in the mortality associated with infection and the impact of the infection on the quality of life of patients. Antimicrobials can also be effectively used when there are severe cases of infection, such as after surgery, for example, and they'll be able to treat the condition quicker. So there'll be a decreased duration of illness and a lower impact on the patient's quality of life. There'll also be decreased discomfort for patients because antibiotics usually come with a lot of side effects that are not particularly pleasant. And there would be a reduction in the unnecessary prescriptions of them. There'd be increased documentation of the treatment provided for bacterial and all types of microbial infections, which allows for accurate tracking of resistance patterns and symptoms that can be used to evaluate current local and national policies surrounding antimicrobials and where to introduce newly developed antimicrobials into practice. There's also lower costs for the NHS and patients, as money is not spent on purchasing medications that may not be effective for their health problems. And education increases the awareness in the population, so there may be less disruption caused to patients' lives when waiting for appointments for self-limiting conditions. These are conditions that will resolve on their own given some time. And lower waiting times in clinic, so more serious non-communicable conditions can be treated faster. Improved sanitation and hygiene reduces the spread of communicable diseases, which would then also reduce the burden of ill health. Yeah, thank you.
Hi everyone, I'm Tasnim and I'm just going to be going over the ethical and legal issues that comes with antibiotic resistance. So just to briefly recap, antibiotic resistance is when bacteria evolve so antibiotics stop working, and this creates major ethical and legal challenges. One important ethical issue is the conflict between the individual and the public good, sometimes called the tragedy of the commons. Giving antibiotics may help one patient now, but overuse contributes to antimicrobial resistance, otherwise known as AMR, which harms society in the future. This also links to antimicrobial stewardship, as mentioned earlier, which means using antibiotics responsibly to preserve them. There is also a conflict between patient autonomy and non-maleficence. Patients might want antibiotics even when they are not needed, like for viral infections, but doctors have a duty to avoid harm, including long-term harm caused by resistance. Doctors also follow beneficence, meaning they must act in the patient's best interest. So there is a conflict evident here. Another ethical issue is justice. Antibiotic misuse is often higher in wealthier countries, but resistant infections cause more death in lower income countries. There is also intergenerational responsibility, meaning we have a duty to protect antibiotic effectiveness for future generations. As mentioned earlier, an example of this is MRSA, and MRSA spreads partly because of antibiotic overuse and poor infection control in the past, which led to stricter hospital hygiene rules and prescribing laws in the UK. Legally, governments use public health law to regulate antibiotic prescribing and restrict over-the-counter sales. There are also environmental and agricultural regulations to control antibiotic use in farming. Because resistance spreads globally, international cooperation through organizations like the World Health Organization is essential. Overall, antibiotic resistance shows how individual healthcare decisions can have global consequences, and it matters quite a bit.
Okay, thank you everyone for listening. I'm going to send a link in the chat with the form to fill in if you want a certificate. But other than that, you're free to leave.